everyone. So our group research on Fibrolamellar hepatocellular carcinoma, abbreviated as FLHCC, is just a juvenile liver cancer disease. So kind of an outline of what we're going to talk about throughout the presentation. The first is who's on the team. The second is what is FLHCC. The third is our hypothesis. The fourth is our lab breakdown and conclusions. And the fifth is what actually happens in lab. So now we're just going to introduce everyone that's on the team.
Okay, hi everybody. My name's Ailey. I'm a sophomore. I'm majoring in biology and minoring in chemistry. I have future plans probably along the lines of med school, something to do with oncology. And then a fun fact about me is that I am allergic to almost everything.
So my name is Hannah. I am a senior. There's an ish there because I'm coming back to Concordia for my master's. So currently I have two majors in biology and psychology, and I want to go into behavioral genetics. Fun fact about me is I'm on the track and field team where I throw heavy objects.
And hi everyone, my name is Katrina. I'm a senior and I'm majoring in biology with a minor in chem. My future plan is to go to medical school. Fun fact about me is that I like plain water.
Hi, you guys. My name is Sarah, but most people call me Stick because of my last name being Stickler. So most people won't remember that. My name is Sarah. So I'm a senior majoring in biology, and I have a chemistry minor. Some future plans: I'm going to be in a gap year in the next 2 weeks here, and then hopefully med school, taking my MCAT soon. And then a fun fact about me is, as of like 2 hours ago, I accepted a position as a medical scribe. Woo! So I'll help you out if you need help.
Hey guys, my name's Tim. I am a senior majoring in chemistry. Just got into pharmacy school, which is super exciting. Thank you. One fun fact about me, which probably isn't really super fun, but is kind of more sad than anything, is that I haven't taken any of the upper level bio courses, and here I am in research about all these bio things.
And thriving.
Mm-hmm. So now we're going to break it down a little bit more and actually talk about what FLHCC is. So there are three main background points we're going to go over, which is histology, how it's set apart, the chimera, which is a really big part of FLHCC, and dysregulation of the proline pathway. So the first thing that we want to talk about is how the histology is different from normal liver cancers. There is all sorts of collagen banding. As you can see in that bottom picture with the tumor, it's that thick pink band. That's not supposed to be there. That's not what a functional liver is like. And so that is one big part of diagnosis. The second big background piece is there's a chimera. So you can see that we are looking at chromosome 19, and there's PRK, ACA, and DNA JB1. They're usually separated by about 400 base pairs, but that section in FLHCC is deleted. And then those two genes that were originally separated are fused together, creating something new called a chimera.
And then the last thing about FLHCC is that certain enzymes in the proline pathway is dysregulated. So everything that's highlighted in the orange box or like the black arrow, they're all pointing towards proline. However, we do not see any free proline. And what's interesting about proline is that it contributes to like the collagen bandings, which we believe is being formed in FLHCC tissues.
Okay, now that we quickly have gone over kind of the background of FLHCC, we're going to jump into kind of our goals and hypothesis that we had for this semester, and then overall what we think about the cancer in and of itself.
Yeah, so now starting with our goal is to understand what at the molecular level is causing FLHCC and how we can treat it. So starting with all this, the main player that we're looking at right now is cyclin E. Cyclin E is super important in the cell cycle. It pushes cells from the G1 to this S phase, which you can see is in that little red box right there. And how this kind of relates to cancer is that, as we all know, when there's a dysregulation in the cell cycle, it will push cells through, and then most of the time that's what causes cancer. Most of the time, not all the time. So in a normal functioning cell, what would happen is this blue line on our graph down here. So the cyclin E would increase, it would get to a certain point, and then it would push that cell from G1 to S, and then decrease all the way back down to 0 and repeat again. However, we found a paper about breast cancer that talks about full-length cyclin E being cut by an enzyme that makes cyclin E into smaller pieces that we refer to as lower molecular weight cyclin E, or LMW. Now, if you're going back to that graph that we were just looking at, LMW would be the orange line. We kind of just shifted to the right so you could see it a little bit better, but it's not necessarily to the right. But the orange line would be, it would increase, push that cell to the next phase, and then it would decrease, but it doesn't decrease all the way down to level 0. There's a constant amount of it, which is not good. So now kind of relating that to what we can see in Western blots, because we can probe for cyclin E. You'll have N, which is normal samples, and then T for tumor liver samples, excuse me. And so the normal, you'll only see the full length, but in tumor, what happens is that you can also see the lower molecular weight. So in that orange box there, you're seeing full length, which is that thick band at the top, but then you're also seeing all these tiny bands at the bottom, which are lower molecular weight. Same for the purple. You're seeing no full length, but you're seeing all lower molecular weight. And all these findings then lead us to our hypothesis.
Cool, so now that you have the hypothesis and the background down, just quickly talk about the hypothesis. So basically, as you know from like proteomics, that the proline pathway was dysregulated. Katrina talked about that like 2 slides ago. And this is producing a lot of proline, which is, as Ailey just said, is increasing that full-length cyclin E. And then it's being degraded by the neutrophil elastase, making those small chunks of low molecular weight cyclin E. This low molecular weight cyclin E has oncogenic functions, which is why we're seeing the tumorigenesis in FLHCC.
All right, so one thing that I thought was important to touch on, so we have 2 paths that we kind of worked on this semester. So Hannah and Katrina worked kind of on the cell model that's on the left there. So basically for this liver cancer, since it is rare and it is only 1% of all liver cancers, there are not very many available people to give samples to us. And therefore we want to be very strategic with how we use these samples. We don't want to waste them and just run useless tests without really making sure we know exactly what we're doing. And so in the past, what we've done before is called a cell transfection, where you make cells and you put this interesting chimera in them that we've been talking about. And we've done that in the past with liver cells. And then this semester we decided to do them with these HEK293, which means human embryonic kidney cells. And so Hannah and Katrina over winter break, they came in early and got these cells ready, loved them, did all the fun things. And then they would give them to us, which Tim, Ailey, and I, and we would run these Western blots. So it's kind of loading them, doing all these fun things that you do in biology. Here's a little kitty for fun. You'd run Western blots, and then you'd get the liver samples for Mayo, then on the right there, and then we get fun results.
Yeah, so now we're going to actually talk about what all happens in lab. So the first thing that we're going to do is kind of explain a little bit about this cell culture, what Stig talked about, this transfection. So this process, we start with cells, they're in a little flask, and we kind of grow them up. They got to mature a little bit so we can do tests on them. After that, we let them grow for a while so they can become confluent. We want one layer at the bottom of the flask. After that, we count them using a hemocytometer. So that means we have to look at all the little cell dots under a microscope and see how many cells are actually there. Once we reach a certain number, after we dilute them, add more dye, we seed them into what is called a 6-well plate. So that's just a fancy little plate that has 6 different wells. There's also 96 well plates kind of deal. And then you let them grow again. So they gotta really flatten on the bottom of that well. And then we add the DNA that we want. When we add the DNA, we have to mix it with this transfection reagent, which we'll talk about in a little bit. You kind of mix those together, add them onto the cells, and then you have to let them sit for a while. It's a very slow process. As you can see, there's lots of time in between. And then once the cells have taken up that chimera that we've been talking about, they glow green. So it's kind of like that Bio 120 GFP lab. We add that onto the chimera so that we can make sure that we're actually transfecting and these cells are taking things up. And then we collect all the cells from that well, called a cell lysate, and we passed that on to the Western blot group to work on.
All right, so this semester, me and Hannah lots of experiments to test parts of the methodology and materials to see what will give us or produce the highest rate of transfection and cell viability. So there are 5 sets of experiments that we did. The first is cell confluency, time collection, transfection reagent, DNA concentrations, and imaging perimeters. So first I want to talk about cell confluency. So cell confluency is just the percent coverage of the wells covered by the cells, and we tested 80% to 120% confluency. So 100% confluency is our standard, just based on past protocols. So the reason why we care about confluency is because if we have a low percentage of coverage, that leads to a low success of transfection. High percentage leads to a higher success of transfection. The next thing that I'm going to talk about is time collection. And time collection is just the best time period to lysate cells from the wells. So we tested 24, 36, and 48 hours. And the purpose is that if we were to collect at a short time period, there would be like a low transfection rate just because not enough cells are getting the transfection. And if we were to have like a longer time period, that kind of leads to like a high rate of cell death. And the next thing I'm going to talk about is transfection reagent. And that is just the amount of lipofectamine 2000 reagent per well. So lipofectamine is needed to help with to increase the efficiency of transfection. And what we tested is 6 to 10 microliters of lipofectamine, and the standard from past protocol was 10 microliter. And through like papers, we found that you should test less. So that's why we're testing like a lower amount. And the reason why we even care about lipofectamine reagent is because if we have a low amount, that leads to like low transfection rate, higher reagent amount, that means that it's toxic to cells. The fourth thing is the DNA concentrations. DNA concentration is just the amount of DNA per well that we're introducing for each well. And what we tested was 1 microgram to like 2.5 microgram. Before, like the old protocol first said that it was from 2.5 microgram to 4 microgram. However, we found that to like kills our cells a little bit more. So after doing some research, we found that we should do at a lower amount. So that's why we were testing 1 to 2.5. And the reason why we don't want like a low DNA amount is because it leads to like low transfection rate, and then high DNA is toxic to the cells, so causes cell death. And the last thing is imaging perimeters. So imaging perimeter is just mainly the setup of the camera and the AM scope software. And for that, we just kind of play around with the exposure time, the tint, and just kind of like the focus to see what would give us the best pictures.
So we put all of that together and made a perfect protocol. Perfect is in quotes because that's still changing. So we found that 120% confluency was the best. That really provided with enough cells to transfect, but not too much that they got overcrowded. And then we decided that 24 hours and 36 hours was going to be our best bet for time collection, because 48, we ended up killing a lot of cells. Sorry, Dr. Yang. Transfection reagent amount, as Katrina talked about, too much is toxic, but too little, we're not seeing enough transfection. So we found kind of a middle ground with 8 microliters of lipofectamine. And then our DNA concentrations, we really worked on a smaller kind of our DNA amount. And as you can see in these pictures, you're seeing lots of little green lit up circles in the bright field images, which is really important because every cell that's lit up has taken up the chimera. And so we're not to 100% transfection rate yet, but that's where we're hoping we can go.
Okay, now we are going to talk about like the Western blot part of research. So I don't know about you guys, but when I was in like Bio 120 and stuff, I didn't know what a Western blot was. So basically a Western blot is pretty straightforward. You start off with just doing normal gel electrophoresis. You do this in like Bio 120 and genetics, and also some of your upper level classes like molecular biology. It's super similar to how you did it with DNA, except for what you're doing with protein. So after you apply an electrical field to your gel. It's going to separate the proteins by size. And then to actually analyze those proteins, we're going to push it onto what's called a membrane blot. And then after that, we're going to put, in our case, we used just normal milk and blocked it off so we could see like the actual proteins that we wanted to look at. Then following this, we put a primary antibody on, which actually detects the protein on the blot. Obviously, the proteins are so small, you can't just see them with your eyeballs. And then following this, we added a secondary antibody, which finds that first primary antibody. And that secondary antibody actually reacts with a substrate that we put on after that. There's like a whole like stepwise thing that you have to get to your final results, which you can see at the bottom of the picture, which is imaging. And there, after we get a good picture, like Hannah's circling, we're gonna do densitometry, and that's just gonna quantify how much protein we actually have in each one of our lanes.
Okay, so now moving up to some of our results from this past semester. So on the left right here, you're seeing one of our blots that we did one of the first couple weeks. And these all are samples from the liver. So at the top, we have the titles of each sample, and N at the beginning means normal, and T at the beginning means tumor. So this red box here, around 2 of the tumor samples that we have, shows what we were looking for, which is that top darker band is the full length, and then that faint darker band right beneath it is the low molecular weight that we were looking for. We also ended up doing densitometry for each of the blots that we did, and that bar graph you see right below it shows a quantitative analysis of each sample.
And then moving on to the right half of that screen, so we did, this is our last data right before Corona, so we were kind of rushing to get through it, but we're really glad we were able to get in there one more time. So we did liver samples, like Ailey said, the N means normal, the T means tumor. And then we put on, right next to the ladder there with the colors, we put a HEC-293 sample from Katrina and Hannah. So that was really fun to see. And those are beautiful looking bands, so that was really fun to see as well. And the difference between this and the week 2 is that this is used with a polyclonal antibody. And this means that it has multiple epitopes. So it means that it can connect to multiple different spots on an antigen. So like if you're talking about a human, it would connect maybe to your head and to your toes and to your hands, or maybe the one that Ailey was talking about, maybe it would just connect to your head instead. So that's why we're maybe seeing a little bit more bands too as well there. And then on the bottom, there is some fun little quantitative analysis and those fun little colors on the graph there. Thank you, Hannah. All right, so then for final conclusions, I'll just start with the Western blot on the right there. So based off the Western blots that we're getting and then the quantitative densitometry, we're thinking that there's a difference between the low molecular weight, so those are the littler bands on the bottom, and the full length cyclin E that's in both normal and the tumor. And then some of the inconsistencies between the samples means that we're thinking, it's not entirely proven, but other papers have slightly touched on it, that since these cancers are at different stages, 1, 2, 3, and 4 that you've maybe heard about, that could be the reason why they're producing different amounts of cyclin E, either the low molecular weight or the full length. So that's just something to consider when we're doing some statistical analysis and just general searching for this molecular cause. And then lastly, the polyclonal antibody that I touched on in the last slide, we're thinking that it detects liver samples, especially tumor, in different and more distinct ways than the monoclonal that Ailey was talking about on the previous slide.
And lastly, just to conclude about the sub-model, so like Hannah and what I said earlier is that each of our experiment was used to test one of the property of the methodology to produce like the highest rate of transfection. And what we found is that cell confluency, we need it at a higher cell density, just so then we get a higher rate of cell transfection. Time collection is from 24 to 36 hours. Transfection reagent is 8 microliters of lipofectamine, and DNA concentration is 1.5 microgram per well.
Okay, so we just also want to touch really quickly on all the fun that we actually have in lab. We just showed you all our results, obviously, and those are super interesting and fun. but we also want to show you like we actually have a lot of laughs.
Mm-hmm. Yeah, so there's a bunch of fun things on here. Here's some semester highlights. So on the left was like our first day as a group. Yeah, thanks, Hannah. Super cute. So we went out to lunch together. And then on the bottom left, there's some coffee orders since Tim and, sorry, Kitty's joining again. Tim and Hannah were RAs, so then they were able to buy us coffee with their extra points. So that was super helpful. And they have all of our orders memorized. So shout out to Natalie in the coffee shop for making our orders every single day. And then kind of moving left to right there, Tim and I went to Katrina's performance a little earlier in the semester. That was super awesome. And then us three again, we went to the Capitol last month, right before Corona happened, luckily. And we were able to present at the state Capitol and got to meet some fun politicians and connect with some people in the upper management of the university. So that was really, really cool to practice some leadership there. And then if Hannah wants to click on some of those fun videos. So this, I don't know why, we were making her poster and then we would just chuck markers at the wall and then try to catch them. I don't know how it happened. And then Dr. Harrison came from the next room because he heard us laughing super hard. So he kind of wanted to get in there. So that was funny. And then the one on the top right, this is when Hannah and Tim weren't getting some good image results and we were really struggling. So Hannah decided to lay on the floor, and that happens a lot with all of us. So just kind of a funny thing. And then on the bottom right there was one day where Tim brought me a little Snickers bar and gave me a nice note. So it was fun to have the support in this group and all that fun stuff.
So the lab, though, for us doesn't always work out perfectly, sadly. And we just kind of wanted to show off some of the things that didn't go great because we were talking how every time we watched I Am Science, it just seems like these students are so smart and like you can't really understand it or like how do you get there? But we just wanted to showcase that you're not always just a smart person. Half the time you're failing, half the time things look really ugly and you're calling your research partners at like 1 a.m. You're just like praying Dr. Yang slacks you back in the next 10 minutes kind of deal. And so these are just some of the failures of blots that we had or stains or like you start to think you know things and then you put in the wrong buffer and things just go a little haywire. So that's what we have for you. We just want to say thanks for listening and good luck with finals.
Hope you're having a good quarantine.